This conference will now be recorded.
Hello students. Now are in the 10th chapter. This chapter is coming under botany chapters, cell cycle and cell division. So now we are moving only through botany chapters. So if you are doing like that, you will get a connection between all the chapters. And here in the 10th chapter, we need to learn about the phases of cell cycle, mitosis, and the significance of mitosis. Then we are learning about meiosis and which are all the stages of meiosis. So in meiosis, you will get meiosis 1 and meiosis 2. And here the difference between mitosis and meiosis also you need to learn. So we have gone through all these topics about cell cycle. We have learned the stages G1, S, G2 and mitotic phase. All these phases we have learned. Then in that itself karyokinesis and cytokinesis we have learned. Then the stages of mitosis like interphase is there. From there you're starting prophase, metaphase, anaphase and telophase. And like that we have learned about the stages of meiosis also: prophase 1, metaphase 1, anaphase 1, telophase 1. That is meiosis 1. And in meiosis 2, prophase 2, metaphase 2, anaphase 2 and telophase 2 we are learning. Now we are moving to the previous year questions of this chapter. So here you can see the questions, even the general questions about the cell cycle and about meiosis, specifically about meiosis and mitosis. You can see the questions. So we are moving to the questions. Here the first question is: a cell in G0 phase. And here four statements are there. First options like exit the cell cycle, option B enter the cell cycle, option C suspended the cell cycle, suspend the cell cycle, and D terminate the cell cycle. So which option? A cell in G0 phase. And you know G0 phase, that is the stage in which the cell exists. Or if a cell which is existing in the G0 phase, that cell will exit the cell cycle, right? Exit in G0 stage, exiting from the cell cycle. That is happening. So in G0 phase, that is a stage. In that stage, the cell will exit from the cell cycle, right? Nothing is happening there. So it is exiting from the cell cycle. And that is the stage which is called as quiescent phase or quiescent stage. And in that, the cells do not divide. Okay, there is no cell division, and that is a permanent state for some cells. Example, in neuron cells, you can see such stages permanently. Okay, so here while learning about G0 phase, these many points for neuron cells, this and all we have discussed while saying about the G0 phase. So when it is entering to the G0 phase, that means that cell is exiting from the cell cycle. And here for some cells, it is a permanent stage like neuron cells. Okay, and this stage or this phase is also called as what? This is also called as quiescent phase. Okay, so when we are moving to the next question, the correct sequence of phases of cell cycle is, see that diagram I told you while learning about the diagram should be there always in your mind, about diagram of cell cycle. So here the correct sequence of phases of cell cycle, which is the correct sequence of say cell cycle: G1, then S, G2, M. The first option is the correct option. G1, S, G2, M. First is the growth 1, then it will go to the synthetic phase, then G2 phase, then it is going to the mitotic phase. Clear? So here G1 and G2 that represents first and second growth phase. This is growth phase 1 and this is growth phase 2. Right? Here S phase, which is the S phase? S phase that represents the synthesis phase. Right. So during that synthesis phase, what will happen? DNA replication is taking place. DNA synthesis is taking place. Right. And M phase, after all these, it is going to the M phase, and M phase is the mitotic phase. Right. So here in the mitotic phase, only that phase the cell is dividing. Up to that, cell is getting prepared for the division. Clear? So that's the second question. And in the third question, you can see post mitotic gap phase. Post mitotic gap phase that is characterized by all except option A, synthesis of RNA and nucleotide. Option B, no change in DNA content. Option C, synthesis of histone proteins. Option D, growth phase of the cell. Here four options are there. Post mitotic gap phase that is characterized by post mitotic means after mitosis one gap phase is coming, which is that post mitotic gap phase. After mitosis, next which one is coming? G1, right? Growth phase 1. After mitosis, those cells are entering to the growth phase, right? So growth G1 phase that is characterized by all except one feature. All the other features are there in those post mitotic gap phase. What and all they? See, post mitotic gap phase, we said that is a G1 phase which is characterized by the growth phase of the cell and synthesis of here growth phase of the cell that is there, then synthesis of RNA and the nucleotides, and here no change in the DNA content. The cell growth is taking place, the DNA content is not changing, right? So DNA content change or that synthesis, that is in the synthesis phase, right? So here the DNA content except one thing, that is a synthesis of histone proteins. So this is the answer, synthesis of histone proteins. That is the right answer. So histone proteins and DNA both are synthesized. In which phase? In S phase of the cell cycle. Synthesis of histone protein plus DNA. Both are taking place in the S phase of the cell cycle. Clear? Next question is, a somatic cell that has just completed the S phase of its cell cycle. It's a somatic cell, okay, and that has just completed the S phase of the cell cycle. S phase, okay, as compared to the gamete of the same species that has first option 4 times the number of chromosomes and twice the amount of DNA. The number of chromosomes that will be 4 times and twice amount of DNA will be twice. Second option, twice number of chromosomes and 4 times the amount of DNA. Option third, the same number of chromosome but twice the amount of DNA. Option 4, twice the number of chromosomes and twice the amount of DNA. First of all, here you need to read a somatic cell that has just completed S phase. That is a somatic cell and that has completed S phase. That means the synthesis phase it has completed. Okay. So compared to the gamete of the same species, when we are comparing the gametes of the same species, that cell is having, see, somatic cell, we know that somatic cell, we have mentioned here somatic cell, that means that is having 2n number of chromosomes, right? 2n number of chromosomes and 2x amount of DNA. DNA will be 2x DNA, right? Then a cell which is completing, which is entering and completing the S phase of the cell cycle. Here we are saying about the cell cycle, right? So when it is entering the S phase or the synthesis phase of the cell cycle, DNA content of the cell will become double, right? Here the 2x DNA, which is present in the somatic cell after S phase. In S phase, it will become 4x, right? It will become double. So it's becoming 4x here. So the DNA content of the cell becomes 4x. Why? Due to the DNA synthesis or the DNA duplication. So the number of chromosomes will remain the same, right? There is no change in the chromosome number. This is what I said while writing, while getting questions from this area. You'll get confused. So read somatic cell. Somatic cell is having 2n number of chromosome and 2x DNA. And after completing the S phase, which is that S phase? Synthesis phase. Synthesis phase means what is happening? The DNA synthesis is happening. There is no change in the chromosome number. So if the somatic cell that has undergone and completes the S phase means it will be having twice the number of chromosomes and 4 times the DNA content. So chromosome number will become twice, right? And the DNA content compared to that. So here 4x. After which one? After duplication, the DNA content will become 4x. So here the number of chromosomes remain the same, right? Number of chromosomes, that is not changing. So if a somatic cell which is having 2n number of chromosomes that has undergone and completed S phase of the cell cycle, that will contain twice the number of chromosome and 4 times the DNA content, right? So what will happen? How the gamete will be? So we are asking a question about the gamete of the same. So as compared to the gamete of the same species has. Here, the same number of chromosomes. There is no change in the chromosome number, but twice the amount of DNA. Twice the amount of DNA. So this is the right answer. Okay. Next question. During which phases of the cell cycle, phase or maybe phases of the cell cycle, the amount of DNA in a cell remains at 4C level if the initial amount is denoted as 2C? During which stage? Option 1, only G2. Option B, G1 and S. Option C, G0 and G1. Option D, G2 and M. Right? See, the DNA content. Here we need to say about the DNA content. Here DNA content duplicates during the synthetic phase or the synthesis phase or S phase. This is duplicates during the S phase, isn't it? During S phase only it's becoming double, or the synthesis or synthetic phase means synthesis of DNA, right? And that means here 2n amount, initial amount is 2n. Sorry, initial amount of DNA content 2C, right? So 2C is the initial DNA content, so that will become 4C, right? So the S phase, again after S phase, which phase is coming? So here we learned which is the G1 phase, S phase, then G2 phase, right? So this is in the G2 phase or second growth phase or pre-mitotic gap phase, G2 phase, right? It is the second growth phase. Here the synthesis is happening in S phase. G1, S, G2, M. This is the stages. So during synthesis phase, what is happening? DNA synthesis is taking place and that is going to the G2 stage next, right? So in G2 phase, G2 phase is otherwise called as second growth phase or pre-mitotic gap phase. It is before mitosis, right? So that is the gap phase before mitosis. And in G2 phase, the synthesis of DNA stops, right? Only synthesis is in the S stage or S phase. In G2, it is getting stopped. Okay, so the DNA level remains 4C, where in G2 also it is becoming 4C. In S phase, it's already 4C, synthesis over. And in G2 also, same synthetic content is remaining the same. So in G2 also, it is 4C. Okay, if the initial amount we denoted as 2C, and it is doubling in synthetic phase, that's becoming 4C, and in G2 also it is remaining as such. That's why in G2 phase also. So here, which answer you will write? During which phase of the cell cycle the amount of DNA in a cell remains at 4C level? Here only in G2. In G2 it is 4C level. G1 and S. No, in this G1 it's not in 4C stage, 4C level. Right. In S you can see G0 and G1. In both you can't see this one. And in G2 you can see 4C level but not in M. Right. So here the option only in G2. Okay. And next question, in S phase of the cell cycle, option A, amount of DNA doubles in each cell. Option B, amount of DNA remains the same in each cell. Option C, chromosome number is increased. Option D, amount of DNA is reduced to half in each cell. Okay. Here, in S phase of the cell cycle, what is happening? Amount of DNA doubles in each cell, right? Synthesis of DNA is taking place. So amount of DNA is getting doubled in each cell. Okay, but chromosome number is not changing. Next question is average duration of cell cycle of human cell. That is 24 hours, the average duration. See, if a normal human cell is in a culture, that divides only once. That means once in approximately 24 hours. Daily, in one day, one time it is divided. That means in 24 hours, it is dividing once. So the G1 phase must be that in this G1 phase, that is about 11 hours. Then S phase, 8 hours. And M phase, that is about 1 hour. And some other cells like bacterial cells and all, we know they can divide rapidly. Yeast, bacteria, that and all will divide rapidly in a faster way, right? And here you can see this is the type of question you can expect, which kind of questions are coming from this chapter. Here two lists are there, so you need to match these. Okay, here G1 phase or gap phase 1, that corresponds to interval between mitosis and initiation of DNA replication. G1 phase, interval between mitosis, one interval is happening, and DNA replication that is taking place after that G1, right? Then S phase. What is S phase? S phase or the synthesis, that marks the period during which DNA synthesis or replication takes place. DNA synthesis or replication. Which one? S phase is the synthesis phase. So DNA synthesis means DNA replication that is taking place. Next option is G2. In G2, so during G2 phase or gap phase 2, proteins are synthesized. Okay. So protein synthesis is taking place during which one? During G2. So during G2 or gap phase 2, proteins are synthesized. So the cells are getting prepared for the mitosis, and cell growth is also continuing in that stage. And during G0 phase, which is called as the quiescent phase, in that phase, what is happening? That cell is remaining metabolically active, but it's not, or it's no longer proliferating. Cell is active, but it's not proliferating. Okay. Next question. The centrosome duplicates during first option S phase of the cell cycle, option B G1 phase, option C prophase of the cell cycle, option D G2 phase of the cell cycle. So among this, which one centrosome duplicates? Here, centrosome. What are centrosomes? See, centrosomes, those are the structures which is containing a pair of centrioles and kinoplasm. It's similar to cytoplasm, right? So centrosomes means they have a pair of centrioles plus what? Kinoplasm, right? Kinoplasm means that is centriole or kinoplasm or cytoplasm. That is somewhat similar to cytoplasm, and this divide during the S phase of the cell cycle, right? So division is taking place during the S phase of the cell cycle, and that forms two centriole pair or two centrosomes. Okay, and here you can see during the S phase of the cell cycle, that's the correct answer. And this duplication that is taking place in the cytoplasm of animal cell. Clear? And when we are moving to the next question, during the cell cycle, RNA and non-histone protein are synthesized in which phase? RNA synthesis and non-histone proteins. See, in G2 phase or the second growth phase or the pre-mitotic gap phase, the synthesis of DNA is getting stopped. Right, synthesis is in S stage. So that will get stopped, and then it is moving to the G2 phase. So here the synthesis of DNA is getting stopped, and this particular phase that involved in the synthesis of three types of molecules, which one? G2 phase, that involves three types of molecule, that includes RNA, non-histone proteins, and ATP. So all these things are required for the cell division. It's very important, right? And question number 11. Yeast cell. A yeast cell that can progress through the cell cycle in about options 90 minutes, 30 minutes, 60 minutes, and 120 minutes. Yeast cell can progress through the cell cycle in about 90 minutes. Clear? Next, here one diagram you can see. See one figure. Here you can see here the schematic breakup of the phases or the stages of cell cycle. Right. Which one of the following is the correct indication of the phase in a cell cycle? See, according to this given figure, you can see a figure here, D, that is representing the correct phase. Right? D is representing the correct phase. That means uh, here G1 phase will come. This is S phase and this is G2 phase and this is the mitotic phase. Right? So according to that picture, D is representing the correct phase. That means the S phase or the synthesis phase. Right, synthesis phase of cell cycle. Other options like here you can see option A. What is this? Option A is mitotic phase, A to M. A you can connect M. That means A, that is the mitotic phase here. Up to here it's a mitotic phase. Then B, which is B. B is marked here prophase, metaphase, anaphase. The whole A is mitosis means here B comes prophase, metaphase and anaphase. B will become anaphase. Right. And here you can see in mitosis, karyokinesis is there, cytokinesis is there. Karyokinesis here prophase, metaphase, anaphase and telophase. After that C marks the cytokinesis. Clear? So here exactly marked phase is the, which is marked as D, and that is representing the correct phase, or it is representing the S phase or synthesis phase of the life cycle. See, which one of the following never occurs during mitotic cell division? During mitotic cell division, which one among these four options, which one never occurs? Here, pairing of homologous chromosomes, that will never occur during mitotic cell division. Okay, coiling and condensation of chromatids will happen. Spindle fibers attached to the kinetochore of chromosome, that also will happen. And the movement of centrioles towards opposite poles, that also will happen. Here, which one will never occur during mitotic cell division? Pairing of homologous chromosomes. Okay. Now, we'll go for few questions from the mitosis part. See, all these questions, same topic is coming in different way. Okay. So one question here, anaphase promoting complex. Anaphase promoting complex. Or we can say APC. Anaphase promoting complex or APC, that's a protein degradation machinery necessary for proper mitosis of animal cells. If APC is defective in human cell, what will happen? Okay, so the anaphase promoting complex or APC is absent, or that is a protein, that is a protein degradation machinery which is very important for the correct mitosis of animal cells. If that protein degradation machinery is absent, or it is not working, or it is defective in human cell, what will happen? Chromosome segregation. In anaphase, what is happening? Chromosome segregation is absent, means chromosome segregation will not take place properly. Chromosome segregation will be in trouble. Okay. See, then we said here, which of the following ions are necessary for the assembly of microtubules? For the assembly of microtubules. For that, which ions are important? Mg2 plus and calcium ions. Magnesium ions and calcium ions are important for the assembly of microtubules. So these questions and all came for all India entrance, medical entrance. And we know that it's mitotic spindle. When we are saying about the mitotic spindle, that is composed, mainly it is composed of the composition of mitotic spindle, that is mainly composed of tubulin protein. Okay, tubulin protein. Another important point is during mitosis, this endoplasmic reticulum and nucleolus that will begin to disappear. Endoplasmic reticulum and nucleolus that will start disappearing. At which stage? It is at late prophase or late metaphase or early metaphase or early prophase. Here, during mitosis, this endoplasmic reticulum and nucleolus that will begin to disappear. Okay, this is happening at early prophase. In mitosis, we are saying about mitosis, right? So in mitosis, prophase is the longest one. Which one is the longest one? Prophase. That is the longest phase. And in early prophase, nuclear membrane and nucleolus that will start disintegrating. Here, chromosome number can be doubled by using which of the following? Chromosome number can be doubled by, four options they have given: indole acetic acid, then zeatin, Third one, GA. Fourth one, colchicine. Which is the right answer here? See, colchicine treatment that can be done to the, or that can be double, or that can double the chromosome number, we can say. Okay, colchicine treatment can double the number of chromosomes, and this will not allow the formation of spindle. So these are the things which we need to learn about mitosis. And when we are moving to the meiosis, astral rays. There are some question about astral rays. What are they? See, question is, what are the spindle fibers that connect the centromere of the chromosome to the respective poles called? So those spindle fibers from the pole to that respective centromere of that respective chromosome, that is called as astral rays. And during which stage of prophase 1? In prophase 1, that genetic combination of that parental characters, in which stage we can see that? See, during which stage of prophase 1 genetic recombination? Genetic recombination is taking place. That is in prophase 1, which stage? In Packaging stage. Okay. Longest phase of meiosis. Longest phase of meiosis. Which is the longest phase of meiosis? That is prophase 1. Here, synaptonymal complex, that is formed during which stage? Synaptonymal complex. Synaptonymal complex, that is formed during which stage? See, in zygotine stage, you can see this in zygotine stage. See, what is this? In zygotine stage, you can see a filamentous ladder-like structure, which is a nucleoprotein complex. Filamentous ladder-like structure. That is a synaptonymal nucleoprotein complex. that is called as the synaptonymal complex, and this is observed between the homologous chromosomes. Synaptonymal complex will form between homologous chromosomes. Homologous chromosomes. Okay, and that forms the basis for that pairing and the synapses of meiotic chromosomes. Then another important question from this chapter, almost so many questions while explaining about the topic itself, I marked the important questions there. And next, crossing over, that occurs at which stage? Crossing over. Crossing over. Here, this crossing over occurs at four-stranded stage. At four-stranded stage, right? So it is taking place at the four-stranded stage, and that takes place at Packetin stage. Okay, Packetin stage. See, it takes place at Packetin stage when after the chromosomes have already duplicated, and that is giving rise to four strands after the duplication, right? So after that, these two strands which are belonging to each paired chromosomes. Okay, so here duplication, that is giving rise to four stranded stage, and two strands belonging to each pair of chromosomes. Okay, so that's how crossing over is taking place, and that is taking place in the Packetin stage. Okay, so for all these, for answering all these questions, meiosis, what is mitosis, that diagram should be there in your mind. Meiosis with all stages. Meiosis, meiosis 1 and meiosis 2 diagram should be there in your mind, and the cell cycle diagram. With all these diagrams, you can answer almost every questions from this chapter. And meiosis can be observed in meiosis. This can be observed in spore mother cells. Okay, spore mother cell. You can see meiosis. What will be their condition? 2n. They'll be in 2n form because meiosis shows reduction division. Okay, so meiosis, this spore mother cell will be in 2n condition, and meiosis is a reduction division, right? So after reduction division, or that is a reductional division in which the chromosome number is getting reduced to half. That means chromosomes are becoming haploid, or it will be in N condition, haploid or N. So on the other side, we have got the options. Which are the options they gave? Spore mother cell, that is the right answer because it is in a diploid stage. Tapetal cells, micropores, megaspores, all these. Tapetal cells, megaspores, and microspores, all these are haploid cells. Tapetal cells, megaspore, microspore, all these are in haploid condition. Okay, so there is no chance of reduction or reductional division. Then during meiotic division, during meiotic division, Here we have got 4 options. Homologous chromosomes are separated. Option A, homologous chromosomes, they are separated. That is one option. Option B, here linkage is disturbed. Linkage, that is disturbed. Option C, you will get another option. What I can give, the homologous chromosomes do not segregate. Chromosome do not segregate. Okay, and option D, all of the above. So here, to write this question during meiotic division, what and all sequences of that process you need to learn. See, in meiotic division, homologous chromosomes will pair up, right? During zygote, homologous chromosomes will pair up during zygote stage of prophase 1, right? And through that synapses and during anaphase 1, these homologous chromosomes, each bivalent, that will get separated and that start migrating towards the opposite poles during anaphase 1, right? And this spindles will pull them and that will migrate towards the opposite sides, and ultimately each pole receives haploid group of segregated chromosomes, right? Homologous chromosomes are getting separated. So here the answer is homologous chromosomes are getting separated. Okay, there is no linkage disturbance. Homologous chromosomes do not segregate, nothing like that. During meiotic division, homologous chromosomes are getting separated. And one important thing in meiosis or in meiotic division, in meiotic division, uh, which is the reductional division and which is equational division? See, in meiosis 1, reductional division occurs, right? In meiosis 2, division is equational. Meiosis 1, that is reductional division, and meiosis 2 division is equational division. Here meiosis 1 we can also call as heterotypic division. Heterotypic division, which one? Meiosis 1, and meiosis 2, that is the homotypic division. Okay, that one more name is also there, that also you should remember. Then here meiosis events, how we can arrange the meiosis events, like four options we are getting. First one is terminalization. This is first option. Second option is crossing over. Third option, which one is third option? Synapses. Fourth option, which is the fourth option? Disjunction of genomes. So how to make it in a correct sequence? Here, correct sequence, that is synapses, The synapses, when it is taking place, zygotine, in the zygotine stage, then crossing over during Packetin stage, then terminalization, that is during diakinesis, and distinction of genome, that is during anaphase 1. Okay, so here the sequence is 3, 2, 1, 4. Right, first is synapses, then crossing over, then terminalization, and D, that is distinction of genome, that is taking place in the anaphase. So here, these are the important questions in this chapter. Very, very important questions. All the questions we have discussed here. Few more questions are there which are giving the straight answer. So that is already mentioned while explaining this topic, while giving a review of all the topics. So here this chapter, what you need to do is go through all these topics. Cell cycle. So first what you need to do, you need to learn about the cell. Okay, chapter cell. Then you should each how the cell structure is, you should be clear with that, and the continuation of that chapter is the cell cycle, about the cell cycle and the cell division, and each stage we have learned about cell organelles and all, right? So during which stage what is happening to each cell organelle, and how that and all working, how it is helping for the cell growth and cell division, and how DNA replication is taking place, preparation, how, what and all preparation one cell is doing for this mitosis, all the things you will get from that. Okay, so both the chapters, soon after reading the chapter 8 chapter cell, you should go to the cell cycle and cell division 10th chapter. Okay, then it will be easy for you to find out. See these 3 diagrams. This diagram is important. Then the karyokinesis, here cytokinesis, and this is the mitosis diagram, and this is the meiosis diagram. All the diagrams you should keep in your mind, and each change in each stage, that you should remember very clearly. So there won't be, with diagram, if you're learning means there won't be any confusion. Some extra points are there that also you can learn, that I have discussed, like about some ions and all. So that and all will be very easy for you if you are keeping this diagram in mind. Okay,